Hi, and welcome to another episode of Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Now, today is part 6 and the final episode of my series on cells, and this one is called the dynamic cytoskeleton. Dynamic, that means it's changing, right? There's a lot going on with it. I love this photo of a cell. It's a dyed cell with fluorescence, so you can see the different parts of it, and here you can see that the cytoskeleton is everywhere, and there's different parts of it. So, I say we jump in and take a closer look here. Now, first of all, one of the most important things you need to know about the cytoskeleton is that there are 3 different parts to it. Now, from smallest to largest, we've got the actin filaments. You may also know them as microfilaments. You've got intermediate filaments, also known as keratin fibers, and then the largest ones, which are the microtubules. So those are the 3 components of the cytoskeleton. Let's take a closer look at each one of these. We'll start with the actin filaments. Actin filaments are made up of these subunits called actin. So you can build them up and take them back down again. So actin filaments inside of a cell don't have to be a permanent structure. They can be around for a long time, but they're not necessarily permanent. Now, actin filaments are like rope. You can pull on them pretty hard, but you can't push them. They don't resist compression very well. They have lots of different functions inside the cell, from changing in the cell shape, maintenance of its shape, cell division, muscle contraction. Let's take a closer look here. One of my favorite things to talk about is how muscles work. All animals move by muscle contraction. So muscle is actually a defining character of animals. And they all work in pretty much the same way. You've got these long actin filaments, and you have a motor protein called the myosin. Now here's how it works. Of course, you need energy for movement, right? That energy is coming from ATP. So what happens here is ATP donates a phosphate group to your myosin. You put the phosphate group on, it causes one shape. Take the phosphate off, it changes shape again. And so you get phosphate on, phosphate off. And what happens is the myosin heads walk down the actin filaments and cause them to slide past each other, and the muscles contract. So every movement you make is caused by a muscle contraction with myosin heads walking down actin filaments. Okay, so this is, I think, very interesting, and you'll learn more about this in an A&P class or in a animal form and function class, because there's a bit more to this story. For one, it actually takes a little bit of energy for the myosin heads to come off the actin filaments. That's why when animals die, they get all stiff. It's called rigor mortis, because what happens is you go into rigor, the myosin heads are constantly causing a contraction after death, hence rigor mortis. Now, all cells eventually will divide, right? And you've got the cell membrane. And at the end of cell division, what you have to do is take your cell membrane and pinch it in 2 so you can get your 2 cells to come apart. So imagine you got a balloon. Imagine taking your rope around a balloon and then ratcheting that rope down and pinching the balloon off into 2 halves. That's what happens. So once again, we've got the actin filaments that wrap around the cell, and then we've got the myosin heads that make the actin filaments slide past each other. It gets shorter and shorter and shorter, and you literally pinch your cell in half. So that's where it's used in cell division. Have you ever watched cytoplasmic streaming? Probably not. But you may have seen this and not realize what you're seeing. If you've ever looked at like plant leaf cells or plant cells like Elodea, and you see the chloroplast slowly moving around the edge of the cell, that's an example of cytoplasmic streaming. Once again, you've got these actin filaments along the edge of the cell wall, or technically it's inside the membrane, right? And then the motor proteins are walking the chloroplast and other things around the cell, and that causes the cytoplasm to stream. And that's actually really important for helping move the cytoplasm around and diffusing things. Okay, now that's it for the actin filaments. Let's move on to intermediate filaments. They're also called keratin fibers, and that's because they're made up of keratin. Now, intermediate filaments might have several different types of proteins, and these are much more permanent structures inside of a cell. So you don't really take and build these up and then take them back down again. Once you build an intermediate filament, it pretty much stays there. Here's a good example of one. The nuclear envelope, it's got a double membrane system. It's held, it looks like a bunch of flattened sacs stitched together in a ball, held together by proteins. But on the inside of the nuclear envelope are the lamins. And the lamins are intermediate filaments that help make the nuclear envelope stronger. And that's all I'm going to say for right now on the intermediate filaments. I know, not very much, but that's all I'm going to say for now. Because I want to talk about microtubules. Now, microtubules are made up of alpha and beta tubulin. There's a plus and a minus end to a microtubule. And like actin filaments, they're made up of these polymers, right? So that means you can build up a microtubule and you can take it back down. So these are not necessarily permanent structures inside the cell. You can move them around to where you need them, but like you have to build them up where you need them. So microtubules have several important functions inside the cell, including cell division and cell motility and movements of organelles. So let's talk about this. When cells divide, whether through mitosis or meiosis, the microtubules are really important. And being a hollow tube, microtubules resist compression. That's a fancy way of saying you can push with a microtubule because guess what? You have to push a cell apart. So there's a structure in cells that forms during cell division, mitosis or meiosis, called a centrosome. And it helps build and organize the microtubules. And specifically when a cell is going to divide, we call it the mitotic spindle. It's basically just microtubules. And what they do is they attach to the chromosomes and they move the chromosomes to the center of the cell, arrange them all in order, and then they pull the chromosomes apart as the cell begins to divide. So these microtubules are very important for arranging the chromosomes. And then another type of microtubule is involved with actually pushing the cell apart. So that's pretty cool. So that's why microtubules are very important. And understanding them, I mean, we're still figuring out how microtubules work inside of cells. And a lot of our cancer drugs target microtubule formation during mitosis, something I'll talk more about when we get to the cell cycle. Now, do you remember in the previous lecture when I talked about the transport vesicles between the rough ER and the Golgi? And I said, these transport vesicles, they don't just randomly move, they actually walk down microtubule tracks. And there's another type of motor protein involved here. Okay, so here's an electron micrograph. You can see these microtubule tracks inside the cell. Here you can actually see the transport vesicles. So let's talk about these motor proteins. We got introduced to one, the myosin. Well, there are two that work with the microtubules, dinan and kinesin. So in these transport vesicles, when they're moving between the rough ER and the Golgi, you have something called kinesin. And kinesin works kind of similar to myosin. It changes shape depending on whether or not there's a phosphate there. So these motor proteins take the chemical energy in ATP and transform that into mechanical energy. And they do that by the ATP donates a phosphate, chain assumes one shape, the phosphate comes off, takes another shape, phosphate on, phosphate off, phosphate on, phosphate off. And these motor proteins literally walk the vesicle right down the microtubule, much like you would walk down a sidewalk. That's pretty cool. Now cells can move around. And one of the ways they do this is they have a flagella or cilia. Now cilia are very short. A cell might have hundreds of them, as you can see in this image here. Or if you have a flagella, they might have 1, 2, maybe 3 or 4 flagella. That's about it. Many cells only have 1. Now the flagella and cilia, like I said, the only difference really between them is their shapes and their numbers, but the way they are put together and function is almost identical. And they're made up of microtubules surrounded by a membrane. So back in the 1950s, we got our first scanning electron micrograph pictures of the flagella and cilia, and they realized they formed this 9+2 arrangement. So basically what you've got are 9 microtubules, and they're doublets, right? There's 2 of them together, hence 9+2 arrangement, and it's called an axonome. Okay, and they run the length of the flagella. Now the question is, how does the flagella actually move back and forth? How is the flagella beating? Well, if you thought motor proteins, you're actually right. This one is called dinan. And then what dinan does is if I've got a microtubule over here and my arm represents another microtubule, the dinan will attach to it. You'll use ATP, phosphate on, phosphate off, phosphate on, phosphate off. And the microtubules basically slide past each other. And then the dinans will release and they'll snap back and they continually, and they beat back and forth over and over again. And that allows the cell to move around. So that's important, right? and understanding how these microtubules might malfunction. Well, some problems with infertility in men comes from the fact that the flagella don't work correctly. Okay, well, that's it for the cytoskeleton, and I hope you learned something, and this is now the conclusion of my series on the cell. I hope you enjoyed it. Stay tuned. Next week, we're going to get into cell metabolism.